Okej, välkomna tillbaka. CEO of Cytovac, Kim is going to take us through the presentation. Just a few words. We are planning an IPO here in Sweden, and we've been out presenting the company for a couple of specialist investors. And the feedback we get over and over is that if you didn't have such compelling data, we would think this is just science fiction. So far so good, and I think they're doing something super innovative, and the data is going to be speaking for itself. So it's a fully enrolled phase 2 asset that you're pursuing, and a pipeline upcoming. Take it away.
Thank you very much. Can you hear me? Yes. Good. Happy to see you all. I can see some familiar faces from our pre-IPO phase in December. I will start by introducing the management team we have here in Cytovac. We have been so lucky that we have been able to hire some very good people in our management team. I'm the CEO, I'm an MD by training, 25 years in the industry, and worked in biotech in many years as well. Our business officer, Finn Eckert Sørensen, has a long history also in biotech, has been 15 years in neurosearch, and Lone has been working with finance in other biotech companies and in pharma companies before she joined us early this year. We've also been able to attract a very science and medical-driven person to our new Chief Medical Officer, and she'll be joining us here 1st of April. What are we doing? We are an immune oncology company within the orphan space. And we have an ongoing phase 2 trial here in Sweden. We have actually completed the enrollment of patients in this phase 2 trial, and I'll come back to that a little bit later. We have also commenced 4 phase 1 trials in different indications. And we have seen early efficacy signals in some of these phase 1 trials. We will publish, or we will at least get our results from the phase 2 trial as early as first half of 2020. That means less than a year from now. And we will expect to have the overall survival data in 2021. Back to what we heard from the previous speakers, it's important to look for the market. It's important to prepare yourself for commercialization, even that you are very early stage as we are. So we have actually looked into the market, and we have done our homework, and the market size we have seen just for the brain cancer study that we are doing is in the size of $300 million, which is still an attractive market, even for Sobi when that time will come. If we should look at our pipeline, you can see that our main indication right now is the glioblastoma, which is brain cancer. We are in phase 2, as mentioned earlier, and we are planning to go ahead with a phase 3 trial after we get our results from that. If we look for the next indication, that's the sarcoma, which is a connecting tissue cancer. This is also an orphan indication. And we have many good reasons to pursue that indication. We are planning to start a trial first half of next year. The triple-negative breast cancer is an investigator-driven study that we would like to support. We would like to support that study because if you are aware that metastatic triple-negative breast cancer patients, which is normally young women, only have an average survival of 18 months, there's a big need for new treatments in that disease. And we have committed ourselves to support an investigator-driven trial in this area. The 2 other indications you can see here is the prostate cancer and the pancreas cancer. And as you can see, we have left that for partner explorations. We have performed phase 1 trials, but we think that these 2 indications are not for us to follow right now. What are we doing? What is our technology? Well, we have a unique technology, but we are not stupid. And I'll come back to that statement in a while. If you look here, what we are doing is that we take a blood sample from the patients, 200 milliliter, and then we bring it to our lab facilities. We have a GMP lab facilities in Copenhagen. and then we actually separate the blood cells into immune active cells. And what we then are doing is we are taking some of these T cells and we develop them into specialized T cells, which is in this case T helper cells or CD4 plus cells. Then we add an agent that would expose a lot of antigens from the DNA in these cells. So now they are exposing a wide range of antigens called cancer testis antigens. And then we add a new pool of the patient's own lymphocytes to these antigens. And now they can recognize all these antigens. And by recognizing these antigens, we can then proliferate these T lymphocytes, which now have become killer soldiers, and then we can bring them back to the patients to kill the cancer. So that is in all fairness what we are doing. I can see some of you are looking a little bit with a question mark. If you have a question, you can raise it also afterwards. Having said this, I would like to say, how are we positioning ourselves into this immune oncology landscape? Because you have heard about immune oncology, and you know that some of these products have been registered for instance, leukemia or blood malignancies. So where do we fit in this landscape? You have heard about checkpoint inhibitors. Maybe you know about antibodies. Maybe you heard about dendritic vaccines. But we are in this field of adaptive cell therapies. And this is a very recognized field where you are focusing on T cells, meaning the active lymphocytes that are going to do the job by killing the cancer. And if you look to the left here, you can see one of the most famous in this group is the CAR T, which has been approved both in Europe and US. What are they doing? They're taking a T cells, a lymphocytes, and then they're doing a genetic manipulation so that these T cells have a target to hit one antigen. And that's very clever if you're talking about blood diseases or blood cancer, because they only have one antigen on the surface. So that's a very good thing. What is the downside of this? The downside is that sometimes it shoots the wrong cells. Sometimes it kills the normal cells. And if you do that, the patient also risks to die. So there are a high safety concern in this respect. The one below is called TCR. And the TCR is actually the same genetic manipulation, but with a much simpler protein. There's a lot of different companies that are pursuing this technology, but it's also a single antigen method to actually hit an antigen in the tumor. They are having the same problems as you can say CAR T have. You need to put the patient's immune system down, because if you don't put the immune system down, the immune system will actually kill these cells, and it would not work at all. So there are some plus and minuses with these technologies. If you look for what we call polyclonal, meaning that you are addressing many antigens or more antigens, then in that group you will see there's the TIL part here, which is a tumor infiltrating lymphocytes, which means that you take out T cells from the tumor, take a part of the tumor, isolate the T cells, proliferate these T cells, and bring them back to the patient. This treatment also has to have a lymph depletion or immune suppression. So you are facing the same side effect as you do with the other stuff. It has never really proved an effect besides melanoma. And maybe one of the reasons is that the T cell you take out for the tumor are tired or exhausted T cells. That brings us back to the last one down here, which is called autologous T cells. Autologous actually mean that you take it from the patient's own blood. That is exactly what we also are doing. What is different between them and us? You can say that this company, for instance, Marker Therapeutics, they take dendritic cells, put few antigens on that, and have the T cells exposed for that. But otherwise, they're doing the same procedure as we are doing, meaning taking blood for the patient, exposed for these few antigens, and bringing these T cells back to the patient. What we're doing is what I just told you, But the difference here is that we expose them for a broad range of antigens, meaning that it could be up to 100 different antigens. And why is that clever? Why do we think it's clever? We think it's clever because all solid tumors express cancer-testis antigens in a different variation. And there's more than one antigen on solid tumors. So only to have one antigen and one T cell to attack this tumor is not what we think is a successful way. So this is one of the big differences from where we differentiate from the other T cells approach that are on the market. Another very important factor is that we do not kill normal cells. And we do not that because cancer-testis antigens are not a part of the normal body. It's not exposed in normal man. It's only exposed if you have a cancer. So that means that I could take a blood sample, hopefully, from most of you, and bring it back to you, and nothing would really happen. And that's a very, very good thing because it gives us a very, very strong safety profile. If we look at what I've just said, these cancer-testis antigens, we are not the only one looking at cancer-testis antigens. Some of the other, if I go back, I hope I could go back. Some of the others here, they also put cancer-testis antigen into this genetic manipulation, but again, just one antigen. So if we look at different tumors, our product can be used for many different solid tumors because these antigens are present on many of these tumors. Why have we chosen glioblastoma, brain cancer, and sarcoma? We have chosen those two indications because there's a high medical need in these indications. It's very aggressive cancers. And also because we think it's clever to use a small orphan indication to actually show an effect. And if we can show this effect, many will be triggered to use it for bigger indications, which will demand much more money and much more muscles, meaning that we will have to partner that. If we should look at the value creation and the flow of news going forward for a potential listing here in a couple of months, for the glioblastoma, clearly the first really milestone will be the progression-free survival data that we will have the first part of 2020. Going forward with that, and that's a part of the commercialization discussion that were earlier today, how do you prepare for that? One of the very important things to prepare for is the manufacturing. If you're not able to manufacture your product, forget commercialization. And this sounds very simple, but there's a lot of big pharmas that have done that mistake. And we have actually worked the last year to focus on our manufacturing optimization, and we are still doing that. And we hope within the next year to have a production which is able to be scalable, which is able to be a tech transfer to a contract manufacturing organization, so that this product actually can be produced anywhere else in the world. Another step for commercialization is that you actually ask the regulatory bodies, what do they want? And that was also back to what we have heard, meaning asking for advice, asking for how do they see our next study, the phase 3? How should that be done in order to get the highest probability of success, but also the highest probability of registration? So that's a very important part that we will work with in the next year. And then, of course, if everything goes well, we will start the phase 2 trial in 2021. At the sarcoma part, as I mentioned, we will initiate a study in next year, early next year, and we will, of course, also, as I mentioned, support the triple-negative breast cancer study, which will actually be started during the summer this year. The protocol is in writing at this moment, so we expect to submit it in May, and then we will have an approval to start this study later this year. If you look at the regulatory news flow, what is the options we have when we get our first clinical results, which will be less than a year from now? Well, we have actually, as we can see it right now, 3 opportunities to proceed. First of all, if it's very, very convincing data, we will take the opportunity to discuss with the European authorities and say, hi guys, could we have a registration based on that? And then submit our overall survival data on a later stage. That would be a natural step, at least to take the discussion. I'm not promising you a registration here, I'm just saying that this is what we want to pursue. And then of course we can plan for a phase 3 trial based on the progression-free survival data, or we'll have to wait until we get overall survival data in 2021, and then plan for a phase 3 trial based on this. For those of you who do not know about glioblastoma, it is a very aggressive brain cancer. The average survival is 15 months, and there's about 34,000 patients in US and top 5 EU. If we go to the local part in Sweden, there's about 400 new patients every year in Sweden. The diagnosis is done by MRI, it's done by scanning, and it's done by a biopsy, meaning that you are actually trying to remove some of the tumor. After having removed some of the tumor, the standard treatment is that you get radiotherapy and you get chemotherapy. But still you do not live more than 15 months on average. And as I said earlier, the market we have calculated here is actually a conservative market with a realistic price. This $300 million is actually only 8% of the total market. This is a very busy slide. You don't have to read all of it. It's just to show you that we have very convincing phase 1 data in 4 different indications. We have 2 in prostate cancer, have 1 in glioblastoma, and we have 1 in pancreatic cancer. And the take-home or take-away message here is that it's very safe, it's well tolerated, we do not need immune suppression, and we saw efficacy signals in both glioblastoma and prostate cancer. I'll come back to the efficacy signals in glioblastoma in a moment. So all primary endpoints regarding safety were reached. What did we see in the phase 1 in glioblastoma? Well, we saw that the 10 patients in our phase 1 study that actually got 3 or more treatments of our product actually had the 7 of those had actually a partial response. So you have to remember that these patients were patients that have been through all other treatment and were progressing at that point of time where we actually did the phase 1. 2 of those 7 actually had a complete response. How is that transferred into a survival? As you can see on the curve behind you here, that these 10 patients had, of course, a different survival rate, but there's a clear tendency to prolong survival. I know this is phase 1 data. I know you cannot use it for statistical part, but still it's very intriguing to see these survival curves. We have made publications, one of them with Cytovac, who have actually shown that there is a very strong dose response with our product. It's always nice to show that there's a dose response, meaning that you can see efficacy and you can see that it depends on the number of T cells that are in our product. Our whole technology and also our phase 1 data has been published in Nature Communication, and you can read all about it there. The ongoing study is we have enrolled 62 patients in this study, and that means that we have completed the enrollment on these 4 centers in Sweden. And the 4 centers you can see on the bottom of this slide, which is the well-known university hospitals in Sweden. The endpoint here, the primary endpoint, is progression-free survival, and the secondary endpoint is overall survival. We need 47 events, and an event is defined as death or progression. And that's for statistical reasons, we need this to actually show statistical benefit. The results are, as I mentioned, within 1 year we'll have progression-free survival, and a year later we'll have overall survival. The comparator in this study is the standard of care therapy. That means it's radiotherapy and chemotherapy. The randomization, we could not do this blinded because it demands a blood test, it demands a donation, and it also an administration of blood, but it was randomized in a 2-to-1 with 2 in the active arm. Back to the market, you can see here that even for the an orphan indication as glioblastoma, there's still an attractive commercial market. And if you add on the other indications that we are looking at, you will see here that they are very attractive markets. And that means that we are also able to attract partners for this, since the market size are in a range where everybody would like to be if the product works. The board, we have a board which have the competences, we have broad competences within our industry. It ranges from biotech competences to being in listed companies. It will cover development, clinical development, regulatory, and also the finance part for the last two parts, the last two members. If you look at our shareholders, we have a broad shareholder base, about 280 shareholders in the company. And in this top 15, you will find an equal distribution of Swedish and Danish shareholders. The biggest shareholder is from Dacco, the Dacco family. You will find Adam Dahlberg in this group, as not in the Dacco group, but in the top 15, and his family, and they are well known for the Gambo family. So if we should look at value over time for the shareholders, this is how we intend to build value for the shareholders, focusing on our orphan indication on brain cancer and bring that to clinical result within very short time. Initiate the investigator-driven study with triple-negative breast cancer. Add on the sarcoma study in 20, and then at some point of time, either partner or expand with a phase 3 trials in these indications.